Hello and welcome to this lecture on second messengers in Wipp's biochemistry. Now, what are second messengers or secondary messengers? They are intracellular signaling molecules released by the cell. Intracellular means inside the cell. Intracellular signaling molecules released by the cell in response to first messengers. Now comes what are first messengers? They are extracellular signaling molecules. That means outside the cell, extracellular signaling molecules. For example, molecules like hormones, neurotransmitters and the like, which we generally call ligands also, that attach, these molecules attach to the receptors, specific receptors on specific cells, so as to carry the message to that particular cell. And inside the cell, based on this message brought about by first messenger, the second messenger triggers or initiates certain processes that bring about changes that affect the cell. So they bring about changes inside the cell, such as cell proliferation, cell differentiation, migration, survival, apoptosis, depolarization in the case of nerve cells, and so on. Now, thus, second messengers form one of the triggers of intracellular signal transduction cascades. Now, let us see what are the examples of second messengers and how they are classified. Let us see a few more points before we actually go on to see the classification. Now, second messengers are molecules having very short span of life. And these second messengers act mostly by activating or inactivating enzymes. So this is very crucial. And removal of the inducing agent or the induction process. Once when the first message, that process is completed or it is no longer there, the second messenger will be broken down and the entire process will be terminated. Now let us see a pictorial representation for this. First messenger activates membrane-bound receptor. Now what happens is that the receptor is activated on the membrane, cell membrane, receptor is activated and produces an effector. This particular effector, it stimulates secondary messenger synthesis. Now, this comes into action inside the cell now. And the second messenger, just a second. Now, second messenger activates intracellular processes. So this is the cascade, the initiation of the entire transduction cascade. Now, let us see the classification now. Yeah. There are basically three classes of second messengers. Hydrophobic second messengers, hydrophilic second messengers, and some gases also. So that forms the third class of second messengers. Among hydrophobic second messengers, we have included just two examples only there, that of diacylglycerol, DAG, and phosphatidyl inositol. We will see PIP2, that is actually derived from phosphatidyl inositol, PIP2. That we will see. Then hydrophilic second messengers are cyclic MP, a very important one, cyclic GMP, inositol triphosphate, IP3, which actually is derived from one of the hydrophobic messenger, namely phosphatidyl inositol. So IP3 is derived from phosphatidyl inositol. Then calcium ions, they are also included under hydrophilic second messengers. Now, the gases inside the cell which act as second messengers include nitric oxide, hydrogen sulfide, carbon monoxide. So all of them are having specific roles in specific cells, and it all depends on the message brought about by the first messenger. The first messenger comes to the cell, and only if a receptor is present on the cell. So it is very cell specific. The message, the transmission by the first messenger itself is very cell specific. So then from there only, once when the message is passed on to the cell, from there on second messengers take charge. So that's how second messenger works. The second messenger acts like this. This is the signal. It may be a hormone or a neurotransmitter. It gets attached to its receptor on the cell membrane. This is cell membrane. This is the receptor. Gets attached to signal. Now the receptor is activated and in turn activates a protein, let us say protein A. This is protein A, which in turn activates another protein, let us say protein B. This is protein B, and protein B gives rise to second messenger molecules. Second messenger molecules like this. So second messenger molecules are not proteins. They are very small molecules. They rapidly diffuse in the cytoplasm, and as you can see, they are produced in large numbers. So its action, or they are amplified. The entire process is also amplified. The second messenger molecules activate another protein. Let us give the name C. That is protein again, and they're actually activated by second messenger molecules. And protein C, once it is activated, it go on to activate the target protein, namely protein D. This is the target protein. It may be a critical enzyme in a metabolic pathway. So it when it is activated, it controls the metabolic process. So this is a classification. You can go through it, and some more points. Just read those lines. And cyclic MP. This is the first example. It's a hydrophilic molecule, and the first messenger of cyclic MP are adrenaline, which is epinephrine, glucagon, another hormone, namely LH. And all these first messengers attach to cell membrane on a particular receptor called G protein coupled receptor. So that receptor gets activated, the alpha subunit gets activated, go on to activate a membrane bound enzyme, namely adenylyl cyclase. Adenylyl cyclase uses ATP to give rise to cyclic MP. And all these things have been elaborately discussed in one of my previous lectures, previous videos, titled Mechanism of Hormone Action. I advise you to go through that particular video to get more idea about cyclic MP and this signal transduction process involved. Now, cyclic MP, during the process of when it controls glycogen metabolism, it induces protein kinase A, which go on to activate the enzymes involved in glycogen breakdown. At the same time, it actually inactivates the enzymes involved in glycogen synthesis also. So this is how cyclic MP bring about or controls glycogen metabolism. Let us see the demo. In the case of cyclic MP, the signal is epinephrine. This is a G protein coupled receptor on cell membrane. This is the alpha subunit. This is adenylate cyclase enzyme. This is cyclic AMP, and cyclic AMP go on to activate protein kinase A, which bring about further actions. Now, during cyclic AMP mechanism, the production of protein kinase A by the action of cyclic AMP. Now, protein kinase A does two things. First of all, it activates phosphorylase kinase by phosphorylation, which in turn activates glycogen phosphorylase, again by phosphorylation. The glycogen phosphorylase activated undertakes glycogen breakdown, that is glycogenolysis. At the same time, as another process, protein kinase A phosphorylates glycogen synthase also. Now, glycogen synthase, when it gets phosphorylated, it gets inactivated, so that glycogen synthesis is effectively prevented. So it acts by glycogen breakdown, enhancing glycogen breakdown. At the same time, it reduces or prevents glycogen synthesis also. Now, so that is a cyclic MP mechanism. Now let us see cyclic GMP. Again, a hydrophilic molecule. Cyclic GMP is synthesized from GTP, just as ATP gives rise to cyclic AMP. This is produced from GTP by another enzyme, guanylyl cyclase. Now, nitric oxide stimulates this process. At the same time, cyclic GMP is a second messenger for nitric oxide also. So, in different conditions, it acts like this. So, cyclic GMP activates another enzyme, namely protein kinase G or PKG. Now, this protein kinase G phosphorylates a number of biologically important molecules, which results in regulation of smooth muscle relaxation, platelet function, then sperm metabolism, cell division, and nucleic acid synthesis, things like that. So effectively, cyclic GMP brings about all these physiological functions also, like vasodilation is affected, retinal phototransduction is affected, calcium homeostasis, neurotransmitters, all these are under the control of cyclic GMP as a second messenger. Now let us see a hydrophobic second messenger. The one here we are going to see is phosphatidyl inositol and phosphatidyl inositol derived second messengers. Now, a derivative of phosphatidyl inositol, namely phosphatidyl inositol 4,5-bisphosphate or PIP2. It's actually PIP2 here. PIP2 can give rise to two molecules, two second messengers. One is diacylglycerol. It is a hydrophobic molecule. It gets attached to a membrane, just as PIP2 also is a membrane-bound second messenger. Just as that, DAG also is a membrane-bound second messenger. And the other molecule formed from PIP2 is IP3 or 1,4,5-inositol triphosphate. So that is a hydrophilic molecule. It gets released into the cell cytoplasm. So PIP2 is actually broken down to DAG and IP3 by another enzyme called phospholipase C. Now let us see a demonstration here. Most of the second messengers are arranged on the plasma membrane like this. Now, adenylate cyclase enzyme here, adenylate cyclase. It uses ATP to give rise to cyclic AMP. And cyclic AMP, the role of which we have already discussed. Another membrane-bound enzyme is phospholipase C, PLC, phospholipase C, and it actually breaks down PIP2 into DAG, that is diacylglycerol, and IP3. So these are phosphates, triphosphate, inositol triphosphate. Now, diacylglycerol being a hydrophobic part of PIP2, it remains attached to the plasma membrane. Now, IP3 can activate or open calcium channels in the plasma membrane so that calcium ions can get into the cytoplasm. IP3 can also mobilize internal stores of calcium by activating calcium channels in internal stores like endoplasmic reticulum or sarcoplasmic reticulum or vacuoles in the case of plant cells. So effectively what happens is that this cyclic AMP, DAG, diacylglycerol, IP3, inositol triphosphate, calcium ions, all these are secondary messengers which play crucial roles in amplifying the signal, the message brought about by the molecules like hormones, neurotransmitters, etc. Here, diacylglycerol stimulates an enzyme protein kinase C, and the stimulated protein kinase C, what it does is that it phosphorylates serine and threonine amino acid residues in certain proteins like calmodulin, and which bring about a number of effects like cell proliferation, regulation of gene expression, etc. So this is a diacylglycerol. And what inositol triphosphate does, we have already seen, it releases stored calcium ions into cytosol. And this released calcium ions will have a variety of functions. Let us see what are the functions of calcium. The calcium in itself is a second messenger. It is normally stored in internal reserves like endoplasmic reticulum. When it is released, we can see at least two functions of calcium ions. Now, one, it activates a number of enzymes. So that is one of the functions. And another one is it binds to calmodulin, which is a protein. And calmodulin, when it is activated, it undertakes a number of functions. Like one of them is contraction of smooth muscle cells. So this is a few of the actions of calcium ions as second messengers. Thank you.